The nanorobot we designed and fabricated is a machine that can be programmed to autonomously recognize target cells and deliver payloads to those cells.
DNA origami is a method that we use in the lab in order to create custom nanoscale shapes that are atomically precise. And the way that this works is that we take a long single-stranded template strand of DNA and combine it with hundreds of short strands that we've designed and ordered from a company. And when we mix these strands together and give them time to self-assemble, at the end we get a custom desired shape.
The nanorobot that we designed actually looks like an open-ended barrel or a clamshell that has two halves. So the two halves of this open-ended barrel or clamshell are linked together by flexible DNA hinges, and the entire structure is held shut by latches or locks that are actually DNA double helices.
In order to load a payload, we need to first attach a DNA strand to the payload, which is complementary to sequences or anchor strands that are hanging on the inside of the barrel. So in a separate reaction, we connect that linker strand to our payload, and then we mix everything together, and the payload is able to diffuse through the open ends of the barrel and bind to the inside.
Now, each of these locks is a DNA double helix, which means it has two strands in duplex. The way it works is that in the absence of the key, which is a molecular protein, the duplexes are held sufficiently strong to maintain the entire structure closed. But when the key is present, that piece of DNA that we designed to recognize that key switches to bind to the key, and the duplex zips open. And when both of these zip open, the entire structure can open. It's very like a combination lock.
So there's a general growing need for smarter pharmaceutical or therapeutics. The amount of information you can pack into a small molecule drug, as they're sometimes called, is quite limited. And so something intermediate in size but still quite smart in terms of its ability to sense its environment, home to the right place and do the right thing is clearly needed. The advantage of using DNA, in addition to being one of the most programmable materials where it's actually predictable what shape it will have based on the computer-aided design, another big advantage is its compatibility with biologicals and human body in particular. And it can be adjusted as to how stable it is, and when it breaks down, it's completely biocompatible. So the potential of this technology is not only in medical therapeutics, but potentially medical diagnostics, where you want to be smart about where and when you're obtaining information, not just delivering payloads. And then there are applications beyond medicine. Basically, any place where you would like to have a smart device that's smaller than electronics, this might be a good candidate.